Hello everyone, welcome back to my YouTube channel. Today we're going to talk about erythropoiesis. If you're new here, please subscribe to this YouTube channel and let's begin. Definition. Erythropoiesis is the process which involves the origin, development and maturation of erythrocytes. The site of erythropoiesis. The site is different in fetal life, infants and adults. In fetal life, we have three stages: the mesoblastic stage, the hepatic stage, and the myeloid stage. In mesoblastic stage, the RBCs are produced in the mesenchyme of the yolk sac. In hepatic stage, the liver is the main site of production of RBCs, and the spleen and lymphoid organs are also involved. In the myeloid stage, the red bone marrow and the liver are the site of production of the RBCs. In infants, up to the age of 6 years, the red bone marrow are the site of production of the RBCs. And up to the age of 20 years, the red bone marrow of the long bones and flat bones are the site of production of the RBCs. In adults, after the age of 20 years, membranous bones like the vertebra, the sternum, the ribs, the scapula, iliac bones and the skull bones are the site of production of the RBCs. After the age of 20 years, the shaft of the long bones become yellow bone marrow because of fat deposition. Hence, it loses their erythropoietic function. During bone disorders, the RBCs are produced in the spleen. The process of erythropoiesis. The stem cells are primary cells capable of self-renewal and differentiation. The hemopoietic stem cells are primitive cells in the bone marrow which give rise to all blood cells. The pluripotent hemopoietic stem cells are hemopoietic stem cells in the bone marrow. There are two types of pluripotent hemopoietic stem cells, the uncommitted PHSCs and committed PHSCs. In the early stage, they are not designed to form any type of blood cells and they cannot determine the blood cell to be formed, hence the name uncommitted PHSCs. Later on, they become designed to form a particular blood cell, hence the name committed PHSCs. There are two types of committed PHSCs, the lymphoid stem cell and the colony-forming blastocyte. The lymphoid stem cells give rise to lymphocytes, while the colony-forming blastocyte gives rise to myeloid cells. Myeloid cells are all blood cells other than lymphocytes. When grown in cultures, they form three colonies, hence the name colony-forming unit E, colony-forming unit GM, and colony-forming unit M. Colony-forming unit E give rise to erythrocytes, colony-forming unit GM give rise to granulocytes and monocytes, while colony-forming unit M, they give rise to platelets. The stages of erythropoiesis. The first stage is the proerythroblast. The cell is large in size with a diameter of 20 microns. The nucleus is large and occupies the cell almost completely. The nucleus has two or more nucleoli and a reticular network. Synthesis of hemoglobin starts at this stage. The cytoplasm is basophilic in nature, and this cell multiplies severally to form the cell of the next stage. The important event in this stage is the synthesis of hemoglobin. The second stage is the early normoblast. The cell in this stage is a little smaller than the first stage, with a diameter of 15 microns. Condensation of chromatin network occurs and the nucleoli disappears. The cytoplasm is basophilic in nature, hence this cell is also called basophilic erythroblast. This cell develops into the next stage. Important event in this stage is the nucleoli disappearance. The third stage, the intermediate normoblast. The cell is smaller than early normoblast with a diameter of 10 to 12 microns. Nucleus is still present in this stage. The chromatin network further condenses and the cytoplasm is still basophilic. Hemoglobin starts to appear, and because of its presence, it stains with both acidic and basic stains. This cell is also called the polychromophilic erythroblast because of the presence of hemoglobin. This cell develops into the next stage. The important event occurring at this stage is the appearance of hemoglobin. The fourth stage is the late normoblast. The cell diameter further decreases to about 8 to 10 microns. The nucleus becomes very small with condensed chromatin network, and it is known as ink spot nucleus. The quantity of hemoglobin increases and the cytoplasm becomes acidophilic, and the cell is now called orthochromic erythroblast. In the final stage of the late normoblast, the nucleus disappears by the process known as kinosis. This cell develops into the next stage. The important event in this stage is the disappearance of the nucleus. The fifth stage is the reticulocyte. This stage is also known as the immature RBC. It is slightly larger than the mature RBC. The cytoplasm contains reticulum, which is formed by the remnants of organelles. Due to the reticulum, it is called reticulocyte. It is basophilic due to the remnants of Golgi bodies, mitochondria, and other organelles. The cell enters into the blood capillaries through capillary membrane from the site of production by the process called diapidesis. This cell develops into the next stage. There are two important events occurring at this stage. The reticulum is formed and the cell enters into the capillary from the site of production. The last stage is the erythrocyte. It is the mature RBC. In this stage, the reticulum disappears and the RBC attains its biconcave shape. The cell size decreases to about 7.2 microns. This cell has hemoglobin but has no nucleus. The two important events occurring at this stage is the reticulum disappears and the cell attains biconcavity. We should note that it takes a total of 7 days for development and maturation of the RBC from the first stage. It takes 5 days from proerythroblast to reticulocyte stage. It takes 2 days from reticulocyte to become a mature RBC. The changes during erythropoiesis. The cell size reduces from a diameter of 25 microns to 7.2 microns. The disappearance of the nucleus and nucleoli. Appearance of hemoglobin and change in staining properties of the cytoplasm. Thank you. That was all for today, and I hope you'll understand the concept. So before we leave, please remember to subscribe and hit the notification bell below. Thank you.